Welcome, everyone. I'm Connor Wander, and I'm here with Maya Gustila, Alzheimer's disease researcher like myself. She's actually more of an expert on amyloid beta, which is one side to Alzheimer's disease, and I'm more of an expert on tau, which is the other side. And there's a bit of a history in Alzheimer's disease where it was previously divided into two camps. And I think at this point, it's fair to say the field is kind of unifying and reaching a common understanding of what causes Alzheimer's disease. So if you want to find out a little bit more about Maya, you can go ahead and listen to her interview. We go over a lot of what brought her into the science field, her motivations, and kind of her journey and how she's been so successful as an Alzheimer's disease researcher and as a scientist in general. So Maya, you're writing a review on Alzheimer's disease, right?
Yeah, I've been working on it with a former postdoc at Ohio State named Holly Brothers, and then also a professor from Australia named Stephen Robinson. Um, and yeah, we're basically trying to show people that amyloid beta may not be all bad as they think it to be.
Very, very interesting. So real quick, I'm going to outline Alzheimer's disease for people who might not be so familiar. So, Alzheimer's disease is one of the preeminent neurodegenerative disorders or neurodegenerative diseases in our world today. It is typically thought of as an age-related disease, although some people who incur brain damage are seeing symptoms very similar to it at much earlier stages. People like who receive traumatic brain injury in the NFL, for example, are getting Alzheimer's disease like symptoms very, very early. And so it's really been in the spotlight lately through traumatic brain injury. But essentially, uh, Alzheimer's disease is caused by aggregation of proteins. Some people used to think that it was aggregation of amyloid beta, and some people argued that it was tau that caused the disease. And what we're really realizing now is that amyloid beta seems to be kind of a trigger to the disease, which causes a bunch of inflammation within the brain. And then tau aggregates because of that inflammation and then causes neuronal loss. And eventually you get brain shrinkage, you have memory issues, you have cognitive deficits, and all of these very profound symptoms that we see in the clinic or in care homes and stuff like that. Really, really devastating symptoms to families and patients alike. And it's a disease that we really don't understand fully. So that trigger and bullet hypothesis is based off of a review by George Bloom. If anyone is interested, I will toss that in the show notes, and I highly recommend it. And it's really been my framework for understanding Alzheimer's disease as a whole. But Maya, I guess you've been doing some research into what causes amyloid beta to deposit and aggregate in the first place, right? And you're really interested in how amyloid beta could actually be protective, and maybe we're kind of looking at the wrong starting point. Maybe there's a different trigger, or maybe the gun is something else entirely.
Yeah, exactly. And so you kind of touched on it as well. For the past, several decades, we kind of thought that the first event that causes you to develop Alzheimer's disease is that you have this accumulation of amyloid beta, and that's called the amyloid cascade hypothesis. So you get amyloid in your brain, and then everything else comes after that. And it was kind of assumed it was just kind of like a random mistake, essentially, that your brain starts to produce amyloid beta. And so then if that's the case, all we have to do basically is get rid of the amyloid beta and everything should go away, right? And it turned out that didn't actually work. And so we've had more than 200 clinical trials, mostly of anti-A beta drugs to combat Alzheimer's disease, and they really just have not worked. We currently have 4 drugs on the market for Alzheimer's, and they don't cure the disease. They slightly improve your memory, but they don't make you live longer. They don't reduce the rate of disease progression. And this has kind of caused people to question this amyloid cascade hypothesis, and think that maybe there is something upstream of amyloid beta, and that by getting rid of it, we're not actually treating the root cause of the disease.
Hmm. Could be making things worse in some cases, especially if amyloid beta is protective, right? So yeah,
Yeah, exactly.
these drugs are like band-aids essentially for someone who's bleeding out of an artery, and it's not enough, and it's often too late. So we definitely need to improve, and that's what we're working on. So a little bit into amyloid beta and how it's produced in the brain. It's actually a piece of a protein that is initially membrane bound, right? So it's amyloid precursor protein, and that's found in all your neurons, and it's very common throughout the brain. So it has normal functions, you might presume. But when it gets cut into these little segments, amyloid beta 1 to 40 and amyloid beta 1 to 42, it tends to clump up. And then that's when we start to see potentially some bad effects in the brain. And so some people thought that it was just the production of this one little protein segment that was really the root cause of Alzheimer's. But recent evidence seems to suggest that there's a purpose to that, right?
Yeah, and a lot of people thought, okay, maybe this precursor protein is doing something, but when it gets cleaved into A beta, that's when it becomes bad. And there's a couple of reasons that we no longer think that's the case. For one thing, amyloid beta is actually really highly conserved, and so like the genetic sequence of amyloid beta specifically, not just the entire precursor protein, is really highly conserved. You actually see it in pretty much every mammal you look at. Even when you go back to flies, they have a version of amyloid beta. In a lot of cases, that A beta is actually necessary. If you get rid of it, their neurons stop working. Um, and another interesting thing is that it seems like you can have a lot of A beta without having Alzheimer's. And so you may have heard of the naked mole rat, which has been in the news a lot lately because of all their crazy anti-aging properties. But actually, what you might not know about naked mole rats is that they have really high levels of amyloid beta all throughout their bodies. It's actually pretty similar to one of our mouse models of Alzheimer's disease called the 3xTG model. They have about the same levels of A beta. And yet, naked mole rats don't get Alzheimer's. In fact, they don't get basically any diseases. They're kind of amazing. And furthermore, there are plenty of people who have A beta in their brains at really high levels and still don't show any memory problems. And so this is making us think that, we have this protein. Most mammals have it. Even some non-mammals have it. And, so why would we still have this sequence if it's just harmful? And so that's what's kind of led people to think that maybe amyloid beta actually serves a purpose. And maybe it's overexpressed in diseases, but you can't just get rid of it entirely.
Perfect. Yeah, that's my understanding as well. So it is important to, I think, retouch on the fact that for a while, the Alzheimer's field was so divided. Some people were saying, no, it's amyloid beta. Other people are like, absolutely not. It's tau that's causing all the damage. And a lot of times it was really counterproductive because these people were sometimes seeming to try to undercut each other and disprove each other. And it was kind of this civil war between the two camps. Now, I'd consider myself in the tau camp, but I definitely understand that amyloid beta can be a trigger for tau aggregation, and it can cause these pathological species of tau, which ultimately clump up and they jam up neurons and they essentially just suffocate the neurons and kill them in a host of different ways, it seems like. And amyloid beta can definitely be a causal element in triggering this pathological conversion of tau. So what I'm studying right now in my lab is how this transition state species of tau, because tau has normal functions too, and if we get rid of it, then we do see some learning impairments in mice. So tau is a microtubule stabilizing protein. Your microtubules are your neurons skeleton basically, and also that's highways. So imagine if you took away the bridge supports on all the highways, they'd fall apart pretty quickly and you wouldn't be able to get shipments of whatever, Amazon wouldn't be able to get you your packages in 2 days. And if that happens in a neuron, things start to fall apart very, very quickly. So tau, we think, one, becomes dysfunctional, and then two, starts to clump up and then just jam up the highways. And eventually the neuron dies and we start to see widespread neuronal loss. But we do think amyloid beta is definitely linked in that process and can be one of those triggers, but maybe it's more of a warning sign of neuroinflammation, which would be kind of inflammation within the brain, like an immune response to something, some sort of infection or something like that.
Yeah, and I think that's a good point that, we obviously know that tau has important functions, and tau is pretty highly conserved across species as well. And so there's no reason to think that amyloid beta would not follow the same pattern. I mean, you don't generally have high conservation of a harmful protein that's only causing harm. So that's kind of what caused people to look into it. And also the fact that in a lot of these clinical trials, as you mentioned, that got rid of amyloid beta, some people had really bad side effects. So, they would do a great job of getting rid of the A beta, but number one, it wouldn't improve their memory at all and their symptoms. And sometimes it would create new symptoms. So one thing that a lot of us found interesting when we wrote this paper is that a lot of people in these trials tended to have a higher risk of infections when they were treated with these drugs, especially upper respiratory infections. And that kind of made us think, well, maybe getting rid of amyloid beta actually makes you at a greater risk of infection. And that was kind of what caused us to develop this hypothesis that maybe actually amyloid beta is a part of the functional immune system, and you can't just get rid of it outright and expect people to get better, because that'll just leave them vulnerable to many kinds of infections.
Yeah, absolutely. And a lot of what actually kills Alzheimer's patients in the end is not really the memory loss or anything like that. It's pneumonia or these respiratory infections actually are the ultimate cause of death in terms of an Alzheimer's patient. Although typically by then they've lost a lot of their cognitive capability, and you could essentially say they're kind of more like a vegetative state. It's really
Mm-hmm.
scary stuff. So I actually would love to loop back to the 3xTG model you mentioned. I'm using the same model in one of my labs as well. So we're trying to bridge the gap between the amyloid beta and the tau camp in labs, and we use mice a lot of the time. And a lot of people use mice for cancer studies and other neuroscience studies. And unfortunately, we do need to use a complex animal. It's a mammal, so it's fairly similar to humans in terms of its genetics and just how the brain works. But this 3xTG model is really cool because it has both amyloid beta overexpressed, so extra amyloid beta, and it has extra tau. So it's got both of those sides. And if we can start to understand maybe this amyloid cascade hypothesis in these mice, then maybe we can get to the root cause of it a little easier and drug those targets, right? So we supply a drug that stops the cascade at the very top of it versus at the very bottom, which a lot of the drugs today are at the very bottom, and that's why they're not so effective. So do you work with the 3xTG model yourself, or is it other people in the lab?
So in the lab I worked in the summer when I was in Switzerland, they did use the 3x TG model. I think primarily they more used the APP model, which basically just develops amyloid beta, because they were more focused on that than tau. Yeah.
Cool. Okay. Yeah, I think it's a fascinating model. It actually takes a while to get these cognitive deficits, is what
Hmm.
we found. And it's like kind of a very subtle model. And maybe that's what we really should be switching to in terms of Alzheimer's disease research, because no one gets Alzheimer's disease in like a severe manner unless it's, like, as I mentioned, some TBI. And that may be some kind of different etiologies, a different mechanism of that happening. But I do think
Yeah.
it's fascinating in terms of this like slow decline that they have.
Yeah. And even the 3xTG model, even though it's much better than things that only simulate tau or only simulate A beta, it still is not a complete representation of humans.
Absolutely.
And so, like, especially things like the neuroinflammation in mice is not exactly the same as it is in humans, and the time course may not be the same. So we're still always kind of looking for a better model that fully captures what Alzheimer's really is, and somehow being able to put that into a mouse.
Right, because unfortunately, mice don't actually get Alzheimer's
Right.
naturally. They don't live that long to begin with. It's like 2 to 3 years for like a normal mouse, even a fully healthy mouse. So, yeah, it's kind of sad we have to introduce these uh, like mutant proteins essentially that are a little more dangerous into the mouse brain, and then we try and see what we can fix. There is actually a relative of a chinchilla. I'm blanking on the species exact name, but it lives to about 5 years. And chinchillas, if you don't know, they're like these little fuzzy rodents, and they're really energetic. But by about 5 years, they'll start to show cognitive decline, similar to Alzheimer's disease. So maybe people will start switching to that. Unfortunately, 5 years is a long time to wait too, and that's something we have to think about in terms of time scales, because nobody wants to spend 20 years in their PhD studies. It'd be really hard to just organize logistically a study of that magnitude and of that scale. And of course, we don't want to keep Alzheimer's disease patients waiting for these new drugs that take 5 years to test in a rodent population. So we really have some problems in the field, and we're looking to answer those. So let's dive right into like amyloid beta's antimicrobial functions. So amyloid beta could actually be protective, as you mentioned, right? And that's really interesting to me because one, we've known for a long time it's part of our genome and it's existed there for a long time. So it's probably not 100% harmful, even if it does harm us a little bit. And it seems like it's kind of an antibody sort of response in some of the studies. I think Rudy Tanzi's lab showed that amyloid beta was clumping up on like viruses and on bacteria that had been injected into the brains of mice and worms, and it was actually saving their brains in a lot of cases from damage from these infections.
Yeah, and that's one of the things that we mainly explored in this paper, which should be out in Journal of Alzheimer's Disease soon, so stay tuned for that. But the hypothesis that actually was created something like almost 20 years ago, by our co-author Stephen Robinson is called the bioflocculent hypothesis.
Oh.
And basically, a bioflocculent is just a fancy way of saying any biological substance that can clump around microbes and cause them to kind of agglutinate and not be able to spread throughout the body. And that's what we think A beta does. And so it's kind of counterintuitive because a lot of people think, well, because amyloid beta can clump up in this way and form these aggregates, it must be bad. But in fact, functional amyloids are actually pretty common in nature. So, for example, some plants have amyloids that they use as part of their immune system. They're found in red blood cells. They're actually found in your teeth. They help to form the enamel in your teeth. They've even found that some peptide hormones that are used for signaling in your brain are stored as amyloids inside of the pituitary gland, so kind of as like just an extra compact form.
Interesting.
And so there's no reason that an amyloid can't be beneficial. And in this case, it might actually be really useful in particular because the amyloid beta is positive charge, and it's attracted to, therefore, negative charge. And bacteria have a negative charge membrane, whereas human cells are zwitterionic, which basically means they have an overall neutral charge. And so this kind of gives this mechanism whereby amyloid beta could be attracted to these microbes and kind of clump around them without necessarily being targeted to healthy human cells. And that's kind of the basis for this bioflocculent hypothesis that's been around for a while.
That's super cool. And so when we're looking at these amyloid beta clumps under a microscope, what we might imagine to find in the very center is a bacterium or some sort of infectious particle that really shouldn't be in the brain at all. So it makes you wonder, how did these infectious agents get into the brain in the first place? Because famously, there's this BBB, the blood-brain barrier, and that's supposed to protect us against anything foreign getting into the brain. So the brain supplies oxygen and the blood vessels supply oxygen and nutrients to the brain through capillaries and the circulatory system, but it's supposed to do it through this intermediary, this middleman. It's the gatekeeper, right? You actually just put a post up on your on your blog about it. And these parasites, which are kind of the guard cells, are supposed to stop things like infectious particles from getting into the brain. So what happens to those parasites, do you think, that maybe they stop doing their jobs as effectively and let some things slip through?
Yeah, and so the blood-brain barrier is this really complex structure. That actually was the main thing I was studying when I was in Switzerland this summer at EPFL, was how the blood-brain barrier might be affected in Alzheimer's and in aging. And so it's this really, amazing structure. It has, it's made of endothelial cells. You have parasites, you have astrocytes, even microglia are involved, and all those things could, in theory, break down. And so, as you mentioned on this article that I wrote about on my blog earlier today, it was basically about this study that was published in Nature Medicine, which found that people with Alzheimer's have about 50% fewer parasites in their brains than healthy people. And as a result, their blood-brain barrier seemed to be a little bit more leaky, and they had a lot more blood proteins found in their brain, which blood proteins tend to be toxic to neurons. So you really do not want those crossing the barrier. And one of the main things we know about the blood-brain barrier in terms of aging is that it almost universally gets more leaky the older we get. And so that allows things like viruses and bacteria to potentially enter your brain when they normally wouldn't be. So, for example, something like 90% of people worldwide have some type of herpes virus. And most of the time, you would never know that you have it. It just kind of hangs out in your blood and in your lymph nodes and doesn't do any harm. But what we're kind of finding is that as you get older and your blood-brain barrier is not as effective as it once was, some of these viruses that could have been in your body for decades are actually able to get into your brain and might start doing some harm there.
And herpes virus actually is a neurovirus, which is really interesting. And it's been closely linked as a risk factor to Alzheimer's disease because it is a neurovirus and it can infect the brain, as you said. So I always wondered how these things would actually get into the central nervous system versus just being in like peripheral infections. So like in a shingles concept or like a chicken pox, right? When we get that when we're kids, it's really painful because it in some degree is infecting neurons. But if that were to happen inside the brain, you would think that would be devastating. It's a lot of inflammation. So
yeah, and it's interesting that we talk about herpes as well because You mentioned that it's possible that A beta plaques might have at the center of it a virus or a bacteria, and in fact, that actually is what people have found in the past. So, for example, there was a study, it was quite a while ago, I believe it was by the Wozniak lab, and they found that something like 90% of A beta plaques in people who have herpes infection the plaques contain herpes DNA.
Hmm.
And so that's not the case necessarily in everyone who has Alzheimer's. But if you are confirmed to have a herpes infection and that infection has reached your brain, it does seem to be the case that you find this DNA inside of the plaques much more often than you would in a normal situation where you don't have herpes. And so this doesn't necessarily prove that the herpes virus is causing these plaques to aggregate, but it is an interesting kind of situation. And they found this not just with viruses as well. So things like Borrelia, which is a type of spirochetal bacteria. I think it's involved in Lyme disease and other types of diseases. They found these also can be found inside of A beta plaques. So it's kind of an interesting observation that these plaques aren't necessarily forming around nothing. They're almost kind of forming a protective barrier around these infectious agents.
Mm-hmm. And one of the main cell types that's thought to clear them is microglia, and microglia are your brain's immune cell. So they're ancient macrophages, essentially, and macrophages are your body's whole primary immune cell, or one of the primary immune cells. And macrophages eat things. That's part of their name, is they swallow and engulf infectious particles or just kind of debris and junk. They clear away a lot of waste that's flowing through your body. Now, microglia are essentially macrophages that got into your brain super early in development, and they establish their own population, and then they kind of become their own thing. And they're regulated by neuronal inputs. They talk to neurons. They talk to astrocytes, which are another supportive cell type in the brain, and they can help with clearing away these amyloid beta and potentially infectious particle aggregates. So we think they're a great target in terms of druggable targets and in terms of ways we might modulate the immune system to get a little better at clearing the amyloid beta without causing damage to surrounding brain structures, for example, because some people think that they're hyperactive and they're just clearing away huge swaths of the brain, and they're collateral damage all over. And then some people also think that they're hypoactive, they're not clearing away enough amyloid beta. So I know there's a lot of debate on that, but I find them super interesting, and they're just such a complex cell type as well.
Yeah, it seems like microglia are involved in almost any neurological disease you look at. They have kind of a hand everywhere.
Mm-hmm. Yeah, absolutely. Like multiple sclerosis, for example, they might be part of the demyelination that happens there. But yeah, they're very hard to study in terms of isolating them as well. So people always say, like, if we know they're so important, like, why don't we know so much about them? The problem is once we take them out of the brain, they sort of switch a lot of their phenotype, a lot of their properties are just switched as soon as we isolate them. So they're really hard to study. And one of the things that I'm doing in lab is imaging them to some degree, and we're trying to figure out some of the things in the brain that can regulate them and either enhance their activity or decrease them, and how that could be applied to Alzheimer's disease. So there are a lot of moving parts, I know, but microglia are the key regulator of neuroinflammation in the brain at our current understanding. So they might detect these amyloid beta deposits and then spew out cytokines, which are basically alarm signals that can be good alarm signals like code red, and then they go into alert mode and start clearing away things, or they can also calm things down by talking to each other. And a lot of this crosstalk happens with neurons, so they're constantly communicating with the brain and trying to figure out where they need to go and what they need to do. And if there's an infection, you would expect that they would definitely be on alert. One of the really cool things about amyloid beta in the news, especially lately, I don't know if you heard much about the amyloid beta blood test. And
Yeah, I did. It's really exciting.
I think a lot of people are excited about that because from my understanding, it's measuring a lot of soluble amyloid beta biomarkers, right? And so there's a difference between soluble amyloid beta and deposited amyloid beta. So this deposited amyloid beta is essentially a protective agent, right? It's kind of clumping up and going after infectious particles. But soluble amyloid beta could also be oligomerized, meaning it's bound to itself. And those oligomers have been shown to be pretty toxic in a lot of studies with neurons. So it's, I think, unclear to me what exactly these differences would result in a patient. I don't know if you have much background on that.
Yeah, I think I don't know too much, as you said, about the difference between the oligomers and the plaques. But what I do know is that there are some people who think that some of the reasons that our amyloid beta drugs have failed is because when we give them to people, they actually start kind of breaking up these amyloid plaques, which increases the amount of oligomers. And the oligomer species actually seem to be quite a bit more toxic
Hmm.
than the A beta that's found in plaques. And so that could be why we're making people worse sometimes with these drugs, is that
Right.
we have all the A beta kind of safely sequestered inside of these plaque structures, and now when you start to release it, it actually just makes them go downhill more. And so I think that's kind of their motivation at looking at the different aggregation states of amyloid beta in these people, and trying to connect that to an Alzheimer's diagnosis.
Absolutely, yeah, and it has been shown that certainly aggregation doesn't seem to be linked to or amyloid beta aggregation, I should say, doesn't seem to be linked to cognitive symptoms too tightly. But then when you start looking at the downstream effects like tau aggregation, which can be spurred by these soluble A beta oligomers, like when we add A beta oligomers to neurons, we start to see their tau making very severe changes. And a lot of these changes are pretty subtle at first, but if you give them enough time, can compound and cause a lot of damage to neurons. So one of the things that we're studying in my lab is how exactly this conversion occurs. In tau, it's super complicated because they have what's called post-translational modifications all over the tau proteins. One tau protein can have potentially hundreds of post-translational modifications. These are like little switches on tau. And if you flip enough of these switches, you can make tau work improperly. You can induce dysfunction on it. And these switches are flipped by all sorts of different proteins within the neuron, within the cell. And it's really hard to understand how these proteins would be activated in response to some sort of amyloid beta oligomer formation, or potentially the plaques are forming and then microglia detect that and they spit out these cytokines as an alarm signal. And just some of those cytokines alone, we've noticed, do seem to cause these changes in tau, where it's flipping of all these switches causes tau to assume a more pathological state, a state that can aggregate more readily. And what we're trying to do now is see if we can kind of break that gap. So either we would slow microglia down, but that's probably not a good idea if they are clearing away infectious particles, or maybe we could stop tau in its tracks right before it converts to this pathological aggregatable formation. So there are a lot of different ways to get at Alzheimer's disease. I think the field has tried a lot of them, but there's still a few more to try, definitely.
Yeah, and I think the key that we're eventually going to find out in the next few decades is that there's probably not going to be one drug that completely gets rid of Alzheimer's. It's probably going to be a combination of drugs. So, for example, one of the things that we hypothesized in this paper is that maybe if you first give people some kind of an antibiotic or an antiviral agent that gets rid of the primary infection, and then give them an anti-A beta drug, maybe that will improve their symptoms. There's also a possibility that maybe if you give them an anti-tau drug and an anti-A beta drug, either at the same time or in a particular order, maybe that's the key. And so I think that's probably what we're going to see happening in the future is combinations of drugs, some that may already be FDA approved, um, trying to see if we can actually get rid of all of the different aspects of Alzheimer's and not just treating one singular protein.
Absolutely, yeah. And now that we understand so much about it, we can hit these drugs at multiple targets. I think Biogen has done some great work in terms of they have these anti-amyloid and anti-tau antibodies essentially. So these antibodies would go in and they'd pick up the tau, and then it would help the immune system clear it. Now, in a lot of late stage cases, I think that's going to be great. But we do need to probably figure out what we should do at early stages when maybe we don't have huge detectable levels of amyloid beta, maybe we don't have detectable levels of tau through this PET imaging that's been in the news lately, positron emission tomography for those interested. And these early warning systems are getting better and better. So the earlier we can tackle Alzheimer's disease, the more effectively we'll be able to treat it, prevent it, and maybe possibly even cure it. That's really exciting stuff that's happening.
Yeah, I think early prevention is probably going to be the key as well. I believe it's Novartis who is currently actually doing some trials with people who have a very rare genetic form of Alzheimer's. So it's basically just have this one rare mutation that basically guarantees you will get Alzheimer's when you get older. And they're testing out some of their drugs with these people to try and see if, and, maybe these drugs have not really worked curing people's Alzheimer's once they have it, but maybe they'll be better as a preventative measure to keep them from getting Alzheimer's. So it'll be interesting to see when those results come out, whether that's actually is an effective technique.
Yeah, fingers crossed for just about every Alzheimer's disease clinical trial. I mean, it's just, everyone's hoping and rooting for them, but so far not a whole lot of success. And part of that owes to the fact that we're testing these things in artificial rodent models, and these don't always translate. So we're always looking for better systems, but certainly early prevention seems to be one of the major keys. So what are some of the things that you think about when you're thinking about early prevention? I think it's never too early to start thinking about it. I would advise that no one ever stresses about it, because you can get yourself into a really terrible vicious cycle if you think, oh no, if I don't do this, I'm going to get Alzheimer's disease. But that's really not the case. There are a lot of risk factors out there. Even if you were to do a 23andMe test to figure out you have like the ApoE4 variant, it's no guarantee by any means, but it is an indication that lifestyle changes would help you out as an individual. And I think those lifestyle changes for me, seem to be one of the more evidence-based and more successful things in terms of warding off Alzheimer's disease. I don't know what your thoughts are.
Yeah, I certainly take steps to try and reduce my risk of Alzheimer's. I personally have never taken the genetic tests. I don't think they're particularly useful necessarily, because you can't really do a whole lot with that information.
Right.
Because you could have this gene and get Alzheimer's, or you could not have the gene and still get Alzheimer's. There's really no way to know. But I think everyone should definitely start taking their brain health more seriously, and trying to reduce their risk of things like Alzheimer's and Parkinson's that we could develop later in life. So things like diet and exercise, you've heard a million times. We all need to eat healthier. We all need to exercise more. But it's not just for your body, it's for your brain too. Like, I've heard kind of the sentiment that I don't want to eat, I want to live a happy life and not eat salads all the time. But, it's not just that your body is going to decay, it's also your brain. You could greatly increase your risk of Alzheimer's by not eating a well-rounded diet, not working out. Another important factor that not everyone talks about is sleep. Sleep is so important for your brain health. That was another thing that we talked about when I was doing research this summer. So they actually had discovered recently that amyloid beta is kind of cleaned up from your brain when you sleep. So your neurons release A beta during the day when they're active, and then as you sleep, your brain actually just literally washes it out of your brain. And so when you don't get enough sleep, there's not enough time to get rid of the amyloid beta. And so your brain is literally getting kind of like dirty, basically. Mm-hmm. And this can happen at any age. It doesn't just happen to older people. We have A beta, me and you have A beta in our brains right now. It's not just something that older people have. And so it's kind of ironic that, so many of us as scientists, especially, don't get enough sleep. We're busy working, studying,
not
in order to get
either.
Yeah, exactly. Yeah. But it's really important for everyone really to, take these things seriously, get enough sleep, even though probably 90% of Americans aren't doing all of these things. Any little step you can take is really going to help your brain. I think another thing also to mention, which you had brought up earlier, is brain injuries. Those really dramatically increase the risk of Alzheimer's, and I'm sure other diseases as well. And you may think, I'm not a football player, I'm not going to get head injury, but most people, the most common cause of head injuries is car accidents. And so even just simple thing like wearing your seatbelt every time you get in a car, and taking the turns a little slower when it's icy out. All these things are really important for keeping your brain healthy, especially as you start getting older.
Yeah, absolutely. So there's this study that I've been kind of watching for a while. It's called the Mending Protocol, and it's a metabolic enhancement for neurodegeneration protocol developed by Dale Bresden at University of California, UCLA, essentially. And it had a small sample size, but pretty profound effects. I'll toss the link in the show notes. And essentially all it is just proper diet and some exercise and good sleep. And like you said, sleep is super important. It's hard for some people to sleep, unfortunately. And if they are developing Alzheimer's symptoms, it can get even worse, actually, because Alzheimer's seems to be able to interfere with circadian rhythm, which is just your body's natural sleep cycle. So that, again, is another vicious cycle. But fortunately, exercise and proper diet can help you sleep longer, and it can help better. So some people thinking, oh, no, all I have to do is sleep, I don't have to exercise. Well, that's not really accurate. Your sleep might not be as impactful if you're not getting the proper nutrients. Also, if you're not getting proper exercise, if you're leading a sedentary lifestyle. So this isn't kind of I'm not trying to wag my finger at people, but you really need to think about this in terms of a practical sense in a whole body, like a holistic approach. And I know it seems very woo woo, but part of this protocol was yoga and gluten-free diet. And it, it, these are seeing results. And these are in people that already have some cognitive impairment. So I know it is hard for people to do it, and scientists are certainly some of the worst offenders at this stuff, right? We want to spend as long in lab as we can. We want to get these results out uh, before the week ends instead of, maybe sometime next week. We want to really push for it, but it is important, especially for my fellow scientists, to think about this stuff and just make sure that you are giving yourself proper time to do all these things, because otherwise you probably won't do them. It's hard to, unless you block off that time and say, okay, tonight I'm going to leave at 6:30 and then I'm going to go straight to the gym. You're not going to do that naturally, right? It's harder done than said.
Yeah, definitely. And I would add as well that if you have some kind of medical problem that's causing you to not sleep as well, especially something like sleep apnea, people with sleep apnea are quite a bit more likely to develop Alzheimer's as well as a lot of other cardiovascular diseases because it does disrupt your sleep so much. And so definitely if you're experiencing something like that, see a sleep clinic. There are a lot of great sleep clinics around. A lot of insurance companies will cover an assessment just because it does increase your risk of all these diseases. And so get treatment for these things. Wear the CPAP mask if you need to. Yeah, take your sleep seriously. It's really important for not just your brain health, but pretty much every aspect of your physical health.
Absolutely. And do limit the afternoon coffees if you can. I know, again, that's easier said than done, but or caffeine, which is the active ingredient in coffee that gets you awake and sometimes makes you jittery and like really hyper, can interfere with your sleep cycles. And caffeine has a half-life of 6 hours. So it takes like 6 hours for your body to clear half the caffeine that's in your system. So if you have a full cup of coffee at 4 p.m. because you need to finish your experiment or whatever, and let's say 200 milligrams, then you still have an extra 100 milligrams at 10 p.m. when you should probably be getting ready for bed. And I would personally find it very hard to sleep if I had that much caffeine in my system. So some people have more tolerance, and everyone's different, but it's also something to think about if you are having trouble sleeping. So let's move on to, I guess, the best treatments right now that we think are in the field, and anything that we're excited about in terms of that. So I did mention Biogen's anti-amyloid beta and anti-tau antibodies. I think for late stage patients, that's going to be hugely impactful. These are people that already have some evidence of heavy accumulation of these proteins. And so by this point, they probably have a lot of extra protein hanging around. And if we can clear some of that away, if we can help the body, I think that's super exciting. And then, like you said, these early stage treatments. Anything else that you're really excited about in the field?
These aren't necessarily drugs that are being tested for Alzheimer's, but there have been a couple of interesting trials where they basically just give people an antibiotic and find that they, in fact, did not necessarily have what we would call a classical Alzheimer's. They actually just had an infection that was disrupting their cognition.
Hmm.
And so, for example, there was a trial, I think it was in Greece. It was a pretty small trial, but they basically took people who had been diagnosed with Alzheimer's, but they all had a type of infection called H. pylori, which I believe is the bacteria that causes like stomach and gastrointestinal problems. Don't quote me on that, but I think that's what it causes. It's pretty common bacteria. And all the people who had this bacteria, when they took an antibiotic, actually had improved cognition, which really never happens in Alzheimer's. You might
Hmm.
sometimes stay the same or get worse, and but you can't really expect to get better. And they also had a reduced risk of death after 5 years.
Wow.
And they've been starting to test some different antibiotics in animal models as well. They're still kind of in early phases. But what I would say, if someone if you know someone who has been diagnosed with Alzheimer's, maybe consider getting them tested for some of these common infections like H. pylori, herpes, HIV, and just see like if they have this infection, see if treatment could actually improve their symptoms, because that could be a really simple and quite inexpensive way of at least improving, if not completely getting rid of some of these symptoms. I think that's a really interesting area of research that we're going to see a lot more studies on in the future.
Yeah, absolutely. And especially with this new understanding what amyloid beta is doing in an infectious particle context, there is no bigger possible source of infection than what's inside of us in our gut right now. So we have so many different types of bacteria, and even fungi, other organisms hanging out in our gut. And for the most part, they help us out. They digest our food, and they do a very good job at that. And we need them, absolutely need them. But in pathological states or a dysbiotic state, these things can get unbalanced. We covered this a bit in our microbiome. It's episode 3 roundtable, and so if anyone's interested, they can go and listen to that. But essentially, the microbiome is this huge like jungle of mix of species, and H. pylori, like you said, can occur pathologically, and it can infect your stomach, causing ulcers. And it could actually be present in more than half of the people in the world. And that's kind of scary because most people won't realize that they have an H. pylori infection. But maybe that is what's happening in a lot of these Alzheimer's disease patients. I actually did a journal club on 2 papers that looked at antibiotic treatment to APP mice. So these mice that are overexpressing amyloid beta. So as part of the intro for that presentation, it was noted that Alzheimer's disease patients do have a different microbiome profile, so a different profile of all these different gut bacteria than their regular normal counterparts. So possibly even rebalancing those microbes could improve a lot of symptoms because these microbes secrete all sorts of short-chain fatty acids, beta-hydroxybutyrate being a very famous one that can not only regulate the immune system, but some of these compounds can even cross into the brain and help out with neurotransmission. So extraordinarily complex system in the brain that we've described in terms of Alzheimer's disease, and throwing in the microbiome into that, which is its own like can of worms, is going to make things more complex. But at the same time, that's a whole different arena that we can start to look at in terms of treatments. So some people are looking at antibiotics, like you said, to rebalance these gut microbes that are perhaps pathological in many Alzheimer's disease patients. If you could follow that up later then with a probiotic, so you kind of clean the stomach out, clean the gut out, and then add in positive bacteria, so helpful bacteria, then maybe you could just, it would be a potential cure potentially. So really exciting stuff, and we're definitely looking into that field in terms of what we can do as a lab to research that, and then also how we can support researchers who are going into that and how we can collaborate with them potentially.
Yeah, certainly. There actually was an interesting paper on that, I think last year in Nature, and they basically found that fruit flies that were a genetic model of Alzheimer's, when they messed up their gut microbiome, they actually had worse progression of Alzheimer's disease. So they went downhill quite a bit faster than the regular flies, which is really interesting. That it shows that even whether or not your gut bacteria actually cause Alzheimer's, they at least seem to be involved in how bad it is and how quickly your cognition declines. And I think that's going to be a big area of research in the future. Your gut is so important to your brain. They've even found that, I think it's from Philip Popovich's lab, they found that giving probiotics to people who have suffered a spinal cord injury can improve recovery. And that shows just how important your guts are for pretty much your entire body, including your brain and spinal cord. So
Absolutely.
definitely the gut microbiome is becoming a big buzzword in neuroscience right now. We're probably going to see a lot more works published on that soon.
Yeah, just about everywhere. It's hard to escape it at this point, but for good reason. I mean, the more we learn about it, the more we realize it has such a profound impact about just anything in terms of cognition to immune regulation. Like, it's hard to escape it at this point. And I think that's for the best because it's been so far understudied as far as that goes.
Yeah, I mean, your body contains 10 times as many non-human cells as human cells. So, in a way, we're more microbe than human. So it
Mhm.
makes sense that they would be so important to our health.
Yeah, exactly. And I mean, not always parasitic, but they certainly can be. And in these chronic disease states like Alzheimer's, it's possible that we have been parasitized, that we have this infection, long living infection in our bodies, and that's slowly kind of eating away at our neurons and probably the rest of our bodies. So some of these things are associated with a broken down blood-brain barrier, leaky gut syndrome. Have you heard of that?
I have, yes.
So that's when the gut lining actually breaks down, and that may be actually one of the first things. In some cases, leaky gut syndrome can actually lead to like gluten intolerance, which is again another buzzword, but these things really are connected in that once things start passing over the gut lining, it's like protective layer in the gut that prevents all these nasty bacteria and microbes from crossing into your bloodstream. Once that breaks down, then infectious particles can get into your bloodstream, and then all it would take is a reduction in parasite number, as you mentioned, which happens naturally with age, some sort of gap in the blood-brain barrier for these things to get into the brain and then cause the amyloid beta accumulation that then kicks up microglial activity or sets off the alarm bells, and then we have this amyloid cascade starting, and tau starts to go pathological, and that could be the whole span of Alzheimer's disease pathogenesis. So it's really hard to study, unfortunately, in the lab, and it means that people need to start thinking very interdisciplinarily. So we need to start talking to each other. We need to start reaching out to labs that have expertise in all these different fields, because it's very hard as a researcher to be good at examining the gut profile or the microbiome profile within the gut, and then examining the integrity of the gut lining as well as the blood-brain barrier, and then looking at amyloid beta and also looking at tau. I mean, I can't do that all in 1 week. So these active collaborations are I think extremely important, and we're hoping to form like a network with scientists that we can start to talk to each other a little easier, and people are really doing that. The internet has been super helpful, but I think we could do better.
Yeah, I think we're finally starting to move past the like A beta camp versus the tau camp and them kind of fighting each other. And now we're realizing probably both things are really important. It's not just caused by a single protein.
Exactly.
And we need to start talking to each other and sharing our research.
So I think for anyone interested who may be a researcher, you probably already know about Alzheimer's Forum. I think it's really, really great resource for me. If you're interested in any of the mouse models that we've mentioned, they have all of these different profiles of a mouse model. It'll tell you when this mouse model gets cognitive impairment or when it starts to lose neurons, according to all this literature. It's a great resource for anyone interested in any neurodegenerative disorder, but it's really, really detailed. So that's Alzheimer's Forum, ALZFUROM.org. And I highly recommend it for anyone who really wants to dig into the literature. And then again, I want to recommend your blog, All Science, ALZ Science, at or alzscience.wordpress.com. And you've really done a great job of giving enough visual help to people to understand some of these processes. And you got into not too much detail. I think it's much more accessible than Alzheimer's Forum. So for the average person who wants to learn a little bit more about Alzheimer's disease, I highly recommend your blog. It's incredible. And looking forward to hearing more about what you're researching in the future.
Thank you. And certainly if any of your listeners have specific questions that they're curious about with Alzheimer's, feel free to shoot me a message on the blog or on Twitter. And I'd be happy to write a post about them.
For sure. Yeah. And myself as well. If anyone wants to know a little bit more about the tau side, you can ask me, and I can always, if I don't know, I will forward the questions on to my boss or someone who's studying that exact thing in my lab. So we really are, I think, as scientists, trying to get the word out there and help people understand diseases better, so that one, they know why our research is important, but also how this found knowledge can help them fight off disease or help their family. And that's all I have for Alzheimer's disease. We can go much deeper into it than we have today, but I don't think we want to bore the audience. Maya, thank you so much for coming on. This has been a terrific opportunity to kind of get to know the other side, the other camp, I guess you would say, about amyloid beta, and just bridge the gap in my understanding. I think I learned a lot just today.
Yeah, thank you. I definitely learned a lot about tau from you as well. So yeah, it's great to be here and talk about this.
Yeah, I can go on for hours, so let's not open that can of worms. But we will toss all these resources in the show notes. And if anyone has any questions, again, feel free to contact us. There's a ton of stuff on Alzheimer's disease today. And just, we want to make sure that people are looking and thinking critically when they think about this research, and potentially reading those clickbait articles. Well, always look for their sources and always look at the studies at the end of it.
Yeah, keep your brain health in your own hands. Don't necessarily trust everything you read on the internet.
That's right. And there are even some like supplements that are claiming they're going to ward off Alzheimer's disease. And some of them are made in China and could have high levels of arsenic. Yeah, that could be even worse. So just be careful. And,
Definitely. If you're a caretaker as well, take care of yourself. Caregivers are at a much higher risk of actually dying than the person they're taking care of a lot of times, just because, it's such a physical and mental strain. So if you're a caretaker, someone with Alzheimer's as well, and you're listening to this, take some time for yourself, seek out resources. It's really important.
Absolutely. Yeah. It's hard to treat someone if you're having issues in your own life. So, and again, that's what scientists do themselves too. We're researching like chronic stress, and then we let ourselves get chronically stressed. So,
Yes.
thanks again, Maya. This has been great.
Yeah, thank you.
Thanks for listening, everyone.